So you can take note of all these things from the recording as well, right? Okay.
Okay.
Now, next we will talk about that what is microsporogenesis. Now, one thing is very clear that the moment we talk about microsporogenesis, we need to understand the starting point and their respective product. And there is a difference between microsporogenesis and microgametogenesis. Here we are talking only about the microsporogenesis. So what is the starting point? The starting point for the process is the microspore mother cell, and microspore mother cell undergoes reductional division to form haploid spore. This is what we call microsporogenesis. But before that, let's try to understand that how the process begins. You know, the process begins by one particular cell, what we call archisporial cell. You can see there, that is the initial archisporial cell. Now, this archisporial cell divides periclinial. Now, can you tell me what is the difference between periclinial division and anticlinial division? Question is both of you. Any idea what is periclinial, what is anticlinial? I'm talking about this.
Periclinial is outside to inside.
Now, let's try to understand anatomically. Suppose this is cell. Okay, let it be that this is a cell like this. Okay, now this cell can divide in two ways. One, it can divide in this manner, in the direction of the arrow, right? As a result of division, what will happen? Another cell is going to be formed like this. Okay, so whenever this division is going to happen, we call them anticlinal. Okay, and what is the net effect of anticlinal? It forms single layered cell. So in the chapter of anatomy, we will discuss that is going to form epidermis. So in case of epidermis formation, there is always going to be anticlinal division, right? But suppose the division is going to occur like this. Okay, so this division is actually called as a periclinial division, right? So in case of anticlinal division, the single layer cell is formed, but in case of periclinial division, what will happen? Multi-layered cells are formed. So understood the difference between these two? Is it clear?
Yes, sir.
Okay. So the archisporial cell divide periclinally to form outer primary parietal cells and inner primary sporogenous cells, right? You can see in the diagram. So this is the parietal cells and this is the sporogenous cells, right? So you see here, the parietal cell is going to form the anther wall, right? That is the important thing, that parietal cells is going to form the anther wall. Okay. And the sporogenous cells form microspore mother cell, which I told you that this is the starting point for the formation of what? The microspore, right? And perhaps we can say that microsporogenesis is going to start from the microspore mother cell. And how the microspore mother cell is going to form? The process begins from the archisporial cell, which divides by periclinial division to form outer parietal and inner sporogenous cells. Parietal cells forms the four-layer anther wall, right? And the sporogenous cells form the microspore mother cell. Okay. And what are the four layers? The epidermis, the endothelium, the middle layer, and the tapetum. One by one, we will discuss the very function and the significance of all these layers. So what I suggest, quickly take a note of it. And once done, let me know. Over. Shall I move?
Yes, sir. Just a minute, sir.
Done.
Okay, done. Okay. Done, sir.
Okay. Right. So that is the flow chart you can see there. Archisporial cell forming primary parietal. Then uh, this is the outer secondary parietal cell, inner secondary parietal cell forming middle layer and tapetum. Then the archosporial cell forming primary sporogenous cells, secondary sporogenous cells, microspore mother cells, and microspore tetrad. So while noting this, you can also note down the ploidy because the question is often asked in the NEET regarding the ploidy. For instance, the question is asked, which of these following structure is haploid or diploid? So you see, archisporial cell is always diploid, right? Primary sporogenesis is also diploid. This is also diploid. Microspore mother cell is also diploid, but microspore tetrad, this is going to be a haploid, right? And this entire structure, right, all are diploid. No haploid structure is found in anther wall. Okay, so that is the only thing what you need to keep in mind. That only the microspore is haploid, rest of the entire structure in case of microsporogenesis all are diploid. Okay, note it quickly. Done.
No, sir. While waiting.
No, no, take your time. Shall I move?
Done, sir.
Okay. Now, next we will speak about the anther wall first. Okay, have a look at this the diagram. So one of the question which is asked in me, the very sequence of the different parts of anther wall starting from outer to inner or inner to outer, right? So first thing is very clear, the outer is epidermis, right? So epidermis is followed by number second is a endothelium, number third is a middle layer, and number fourth is a tapetum, right? This is the four layer we know very well, right? So epidermis is a single layer made up of cellulose. Endothelium is very important. Why? Because it is made up of cellulose and pectin, and it is the pectin that helps in the breaking of the anther. Because the anther wall needs to be broken so that the pollen grains can come out, right? And it is the pectin that helps in breaking of the anther, right? So this is a very important thing what you need to keep in your mind, right? Middle layer 5 to 6 layers. Okay. Now middle layer is 5 to 6 layer, and they degenerate after the maturity of the anther. Another very, very important layer is a tapetum. Now, what is the role of that tapetum? You see, tapetum is the innermost layer, right? So being innermost, it provides nutrition to the developing spores. And how they provide nutrition? The very interesting thing is that they divide by division called endomitosis. And in case of endomitosis, we know very well that the chromosome number gets doubled. And if the chromosome number is going to get doubled, that means more and more DNA is there or more and more chromosome is there that will eventually help in formation of more and more protein, right? So that's how this tapetum is going to provide nutrition to the developing spores. Okay. Another more interesting thing is that they have a ubis buddies, right? So these ubis buddies are responsible for the formation of sporopollen. Again, sporopollen is a chemical that is found in the exine of the pollen grain. You know, pollen grain has a exine and intine. Exine is made up of sporopollen that is very, very tough, right? And this sporopollen is being formed by the ubis buddies, and ubis buddy is actually present in the tapetum. So the two things you must know from neat perspective regarding tapetum. First, it provides nutrition to the developing spore. Number two, it divides by endomitotic cell division and provide the ubis buddies which eventually form the sporopollen. Okay. So understood, both of you, is it clear?
Yes, sir.
Okay. Please take a note of it. Once done, let me know. Over. Shall I move? Once done, let me know.
Done, sir.
Okay. So next talk about that what is microsporogenesis. I've already told you that we need to understand the starting point and the ending point. So here what happens, the diploid microspore mother cell undergoes meiotic division to form haploid microspore, right? So you can see there microspore mother cell. So here it is also microspore cell, right? So, so you see, this question is often asked that which is the last stage of sporophyte generation. So what we can say that microspore is the last stage for the sporophytic generation. But of course, for the male gametophyte, the first stage is microspore cell. Okay, not mother. It is a wrongly written over there, microspore cell, because it is a haploid one, right? So please take note of first stage of male gametophyte and the last stage of sporophytic generation, right? So that is very, very important from need perspective. Okay. So this is the very definition of microsporogenesis. In the next slide, we will be discussing about development of the pollen grains. Okay. Please take a note of it.
Done, sir. Done, sir.
Right. So next we will talk about, as I told you, that what is the next course of development in case of microspore. So we know very well that at the initial stage, all four microspores are attached together. This is what we call the spore tetrad, right? And it is the callus that helps in attaching the things, right? So this at this stage we call them tetrad. After some time, what happens? Callus layer dissolve, right? And how they dissolve? They dissolve by callase enzyme, which is secreted by tapetum. So there is another role of the tapetum. They secrete callase enzyme. So that's why their the very attachment among the microspores is going to be detached, right? So most common, there are different types of tetrad. You can see there, depending upon how they are arranged, right? So this is what we call tetrahedral, isobilateral, right? But you please remember these two things. So in case of dicot plant, what happens? We see the tetrahedral. In case of monocot, what happens? We see the isobilateral. Rest of the very, very specific. No need to memorize from neat point of views, right? So isobilateral are found in monocot, and I told you that tetrahedral that is found in case of dicot, right? So important thing is that there is a callase enzyme that is going to dissolve the callose and eventually freeing the microspore. Please note it.
Done, sir.
Done.
Yes, sir. Yes, sir.
Okay. So now you see, now we will discuss about the pollen grain. The formation of male gamete from haploid microspore is actually called as a microgametogenesis. I told you we need to understand the starting and ending point. In case of microsporogenesis, the starting point was microspore mother cell, ending point was microspore, right? Here what we see, that formation of a haploid microspore, okay, uh, then the male gamete being formed from the microspore. This is what we call microgametogenesis. So we can see there are two stages. One, the stages prior to pollination and the stages after pollination. Okay. So haploid microspore or the pollen grain, as earlier also I told you, represent the first stage of the male gametophyte. So here you see, this is the microspore. So first mitotic division is going to happen, right? And once the mitotic division happened, then the two cells are going to be formed. One is the generative cell, second is the that is the tube nucleus, or what we call the vegetative cells. Okay, so these two things are going to be formed during the first things. Okay, and you know very well that pollen grains are normally released in the two nuclear space. That is important. Okay.
Completed, sir.
Okay. Now you see, have a look at the study of the pollen grains. We need to study the pollen grains. The study of pollen grain is known as palynology, and they have a great amount of taxonomic significances. Okay. Now, the pollen wall consists of outer exine and inner intine. Exine is made up of sporopollen. We have already discussed. That is, it is the we know very well. That is basically the what I mean to say. Uh, the tapetum that helps in the very formation. We have already discussed about that, right? Now you see, the sporopollen is highly resistant. Okay, they are most resistant one, right? And since they are resistant, okay, it is also non-biodegradable. So you can see in the diagram, the outer exine, inner intine. So they have a generative cell as well as the vegetative cells, right? And in between them, there is a cell wall, and that cell wall is actually made up of callus. Now, there is a one concept over here. In case of plant cell, you see, whenever any cell which is found within a bigger cell, their cell wall by default, it is actually made up of the sporopollen. Callus, not a sporo. The cell wall is made up of the callus, as you can see here, right? Okay. So please take a note of it.
Sir,
Yeah, tell me.
so what is vegetative nucellus and generative nucellus, sir?
There are two types of cell that is being formed. Okay. And these two cells, we call them vegetative and generative cells, right?
Okay, sir. Done, sir.
Okay.
Done, sir.
No, please take it.
Dull, sir.
Done? Okay, fine.
Yes, sir.
So this is a pollen grain, right? So UV's bodies of tapetal cells helps in the development of the pollen enzyme. Already we have discussed about that, right? Okay. And the aperture through which actually the pollen tube comes out, right? This is what we call pulpus. You can see in the diagram as well. That is also called as a germ pore, or that is also known by the name of pulpus. Mind it, that is very important from neat perspective. Monocots are monocolpate, but dicots are tricolpate, right? That is the things what you need to keep in your mind. Okay. Then what we see, there is one very important case which I don't think that will be ever asked in NEET, but that is just for the sake of information. In case of a one plant, what we call Hyacinthus orientalis, the pollen grains is going to form 8 nucleate embryo cycle-like structure, right? It was first observed by Nimek. That's why it's popularly called as a Nimek phenomenon. Once again, I'm saying that this is actually not being in my NCERT textbooks, so that is not important. Okay, it will not ask in NCERT, just for the sake of the information. Meanwhile, you can write down these 3 points. That is very important.
Complete.
Done.
Yes, sir.
Okay. Now there is a another interesting phenomena, that is compound pollen grain, right? So pollen grains, what happens in some cases, it aggregates, and after aggregation, right, they form a structure what we call compound pollen grain. So that is reported in 2 family, right? And what is the name of the family? The name of the family is the Orchidaceae and Asclepiadaceae. Okay, you can see in the diagram as well, there is a pollinium. So pollinia is a coherent mass of pollen grains, right, in which all the pollen grains are actually joined together. Okay, and once they join together, right, so during the pollinations, they are transferred together. Okay, so that is the most important features of what we see in case of the pollinia. Okay, right.
Yes, sir.
So in case of orchids and calotropis, what we see, they are transferred as a single unit. Okay, please note it.
Completed, sir.
Completed. Okay.
Completed, sir.
Right. So you see now the sequence of events that is going to occur after pollination. Right. So what we are going to see after pollination, that we will be discussing over here. Okay. So here what we see that uh, the intine grows out through germ pore, because you know very well there is a intine and exine over there. So that's why we are saying that intine is going to grow there, right? So once it grows there, okay, then what we see that there is a the vegetative nucleus is the first to enter the pollen tube, right? So write on this first of all.
Done, sir.
Done. Okay. Next, what we
So,
see? No, Bhavya, done.
yes, sir.
Okay. So next, what we see that the generative nucleus migrate into the pollen tube, and after the migration, what they will do? They will just go for this one. That is, they are going to divide it, okay, and they will form two male gametes, right? And you know, there is a consistent falling of the callus plug. And what is the function of the callus plug? The function of the callus plug that it actually uh, just ensure that the male gamete should remain at the tip portion, right? So that is the very function of callus plug. As the pollen tube keep on going, this the the male gamete will push, and every time the new callus plug is being formed, so that it will prevent the male gamete to be being go to the backward, right? So that is the very function of the callus plug. Please note it.
Tell, sir.
Done.
Just a minute, sir. Position. Thanks, sir.
Okay. So next what we see, this is the entire things that microspore mother cell to microspore is a microsporogenesis, and from microspore to the two male gamete, this is what we call the microgametogenesis. Okay, so this is the popular question that is asked in NEET. So it is often asked that we need to figure it out, that what is the number of nuclear division in microsporogenesis. So mind it, there is a one meiosis, but in case of meiosis, what happens? There is a two nuclear division, right? So that's why the number of nuclear division in microsporogenesis is a two. But the number of nuclear division in microgametogenesis is also two. But suppose if the question is asked that what is the number of nuclear division in microspore mother cell to form male gametes? Of course, the answer is 4. Understood?
Yes, sir.
Please note it.
Yes, sir. Yes, sir. Thanks, sir.
Done. Okay. So this is all for today's sessions, right? So in the next sessions, we will be talking about the mega sporogenesis. So any questions, both of you? Any questions, any doubts you want to ask?
No, sir. Sir, what
Okay,
does
any
mean
questions?
by